So I want to give you a view of what the breadth is of our lab's work, including Parkinson's disease and other diseases and other, I think, interesting projects. So let's get started. So most of you already know this, thank goodness, that stem cells are defined by their ability to make copies of themselves and to become more than one cell type. So anything that does that is a stem cell. However, there are two kinds of stem cells. There are cells that we call multipotent. These are the cells, for example, in the bone marrow, that can self-renew and they can become other cell types, but they have limited abilities. The other type is called pluripotent, and these cells are capable of making every cell type in the body. So they're both stem cells, but they have very, very different properties or different capabilities. If you have questions, please ask them whenever you wish. Okay, so multipotent stem cells include all of the adult stem cells, all of the cells that you're acquainted with, bone marrow, for example. We actually have stem cells in many of our organs. They aren't really very functional. We can't regenerate whole organs in most cases. We have stem cells in our brains, for example, and we can recover from small strokes and other problems like that because we have stem cells in our brains. But most diseases require more than our bodies can actually handle. So this is a diagram that shows you, essentially this is a little tree of all the different cell types in the body, or many of them. And right here is one type of stem cell, bone marrow stem cell, which gives rise only to blood cells and not to the heart, muscle, or skin, or brain. And this you'll be familiar with because we've been doing this since the 60s. Using bone marrow stem cells to replace the blood-forming cells. This is often used for people with leukemia or anemias or people who have undergone procedures that kill the bone marrow cells. You can get a transplant, replace them. But these cells are only capable of making blood cells. Okay? These cells are not capable of making other cells in your body. Pluripotent stem cells can become every cell type in the body. And we're going to talk about two types, embryonic stem cells and induced pluripotent stem cells. These are the original pluripotent stem cells. The induced pluripotent stem cells are the cells that we're going to be using in our project. So pluripotent stem cells, using the same kind of diagram, these cells can give rise to all the different parts of the body, including the stem cells that give rise to other cells. So they're like the stem cells. Pluripotent stem cells come from two sources. One is from discarded embryos that have been left over from in vitro fertilization procedures. These are assisted reproduction procedures. They're done all over the U.S. It's estimated that there are around 400,000 embryos left in freezers that are not being used. So originally this embryo, embryos like this, which are microscopic, you cannot see them. They have only 50 or so cells in them. This one little part here inside this is a 5-day-old embryo, before it's been implanted into the uterus. This little part called the inner cell mass, if you can manage to get it to survive in culture, you can give rise to a pluripotent stem cell line. It has the same genetics as this original embryo. And these cells in this dish can give rise to all the cell types in the body. About 5 years ago, first in mouse and then more recently in humans, a researcher in Japan named Shinya Yamanaka discovered that he could actually convert other cell types into pluripotent stem cells. So the main idea is to get a skin biopsy from a volunteer, and those cells are grown up as what we call dermal fibroblasts. You'll see some of those in the lab from actual biopsies from actual people. And then by doing genetic manipulations, which are improving all the time, we can turn those cells into pluripotent stem cells that are an exact match for this person that the biopsy came from. The biopsy is only 3 millimeters. It's just a little punch. I've had it done to myself. It really doesn't actually even hurt. Just put a Band-Aid over it. In a week, it's healed. And the cells are, I'm showing you this now because you're going to see these in the laboratory. This is a colony of pluripotent stem cells. This is human embryonic stem cells, and over here is another colony, which is induced pluripotent stem cells derived from human skin. And the cells in the background are cells which we call feeder cells. We use them to provide support for the stem cells to grow and survive. So I believe that they're going to be showing you probably mostly induced pluripotent stem cells, which we call iPS cells. So they'll be calling them iPS cells, that's the abbreviation. And these are very small, again, microscopic. There's actually a line here that shows that this much distance across this colony is 0.01 inch. So you can't see them with the naked eye. You'll be looking through microscopes. Okay, so how can stem cells be used for Parkinson's disease? I think you already know more than I do about Parkinson's disease. That's a progressive neurodegenerative disorder. It involves tremors, rigidity, slowness of movement. Most of the cases are sporadic, but there are inherited forms of Parkinson's disease as well. They're rare, but they exist. And the part of the brain that's affected in Parkinson's disease is this little part here called the substantia nigra. These cells are making dopamine, and they essentially are providing control over subtle movements to other parts of the brain. So when these cells are lost, and it takes a great deal of loss, like up to 80 to 90% of the cells, then you start getting the symptoms of Parkinson's disease. And it can be managed by making the remaining cells more powerful, by increasing the amount of dopamine made, or by increasing the effectiveness of dopamine on the target cells. Okay, so the idea of using cell replacement therapy for Parkinson's disease is actually one of the first ideas for cell replacement therapy ever. There was a researcher called Anders Bjorklund, and he started reporting his work in 1987. Their strategy was to take aborted fetal tissue. This is just a picture of the brain of a 6 to 8 week old human fetus. And they cut out this tiny little section here that you can barely see, and then transplanted those tissue pieces to the brain, actually to the target area of the dopaminergic neurons that are missing in Parkinson's disease. And they found that there was a remarkable improvement in some patients, long-lasting improvement. The trouble was that there were a lot that weren't improved, and there were some that actually had really bad side effects, which I'll tell you about a bit more in a second. Okay, so this idea caught on, and in the 90s into the early 2000s, there were actually clinical trials of cell replacement therapies in the US. These are FDA-approved, double-blind clinical trials using fetal neurons. In one trial, 40 Parkinson's patients were transplanted. Some of them either had mock transplants. There was an actual double-blind. The surgeon didn't know whether they were getting cells or not. The patient didn't know whether they were getting cells or not. And the idea behind that in a clinical trial is to make sure there's not a placebo effect, or you can detect a placebo effect. And then, luckily, the people who did not get the transplants and did not improve were allowed to come back for a transplant later. So it was sort of a good scientific study with that additional sort of humanitarian aspect to it. Okay, so what they found was that they found 3 different outcomes from the people who were transplanted. In some cases, they saw no change at all. In some cases, in 25% of the cases, they found reduction of the Parkinson's disease symptoms. So that means out of these patients, about 10 of them no longer needed DOPA treatment. However, they had a problem, and that is that this phenomenon called dyskinesia, which is uncontrolled movement